Hello, everyone, and welcome to our 13th lecture in our course on brain and behavior. And so this is going to be the final lecture in our unit on the senses. So our first lecture on the senses, we talk about the senses in general, what properties govern all of them, as well as the somatosensory system in particular. And then we continued on in our second lecture with our senses of hearing, taste, and smell. And so this is our final lecture, and it's on vision. And so we're going to devote an entire lecture to vision, partially because it's really important to us as human beings. As we'll see, about a third of our cortex is devoted to visual processing, and we're pretty good at it. So there are certain animals that are better at vision than we are, but we depend on it heavily. It is, in many senses, our most acute sense. And so we're going to spend this whole lecture on it. So we're going to talk about vision. And we'll start with the structure of the eye. The eye, of course, is the organ we need for vision to be possible. So we'll talk about the eye physically. What are its parts? What do those parts do? And then we'll talk about function. We've already seen sensory transduction in a number of our sensory systems, and vision is no exception. You have to get information from its sort of native physical form, whatever form of matter or energy it takes, and get that into the form of action potentials. So that's true for vision as well. We're going to see how that works. And the first step in that process, of course, is getting light to the retina. And so we'll see what the retina does with that light. How does it respond to light? How does that information get processed a little bit and turned into action potentials before making its way toward the brain? And then we'll look at visual cortex. And visual cortex has a number of parts. When we talk about our visual world, it's very complex. And so we don't experience the world as a set of disconnected little blobs of light. We think of things in terms of shape, in terms of movement, in terms of objects. And so all that happens in the brain. And we'll see a little bit about how that works. How it can break down, for example. So we're going to start with the eye and the retina and see that there are different cells in the retina, and they have different jobs. So we'll talk about what each of those jobs is, what each of those cells does, and that's going to form the basis for the action potentials that our visual system uses. And again, we have a complex visual world. So we'll talk about how we see things in terms of color, how we see things in terms of movement, how we see things in terms of objects, because that's really what we experience. Even simple objects, we don't think of them as a collection of lines or dark patches or light patches. We think of them as objects, even if you rotate them a bit, even if you change the lighting, even if you change their size. So we have a complex visual world, but our visual system is also very complex to account for that difficulty. And we'll see that there are some deficits. There are ways to fool the visual system. There are injuries and deficits that can occur in the visual system. And to some degree, our brain can compensate for those things. It's not always perfect. It's not always a complete compensation, as we'll see, but it is possible. So we'll start with the eye and the structure of the eye. So of course we have to get light into the form of neural signals. We have to turn that energy, photons, into action potentials. So how do we do that? Well, we'll get to the function here in a minute, but let's look at the path that light takes. So light is coming in from the environment. Our eyes don't generate light. They are not like flashlights. And that sounds sort of obvious, but for centuries, people thought that the way the eye worked was by literally shining beams of light out into the world and getting the reflected images back. Sort of like a dolphin would use the echo of sounds that it sends out to figure out where things are. That, of course, is not how the eye works. It is completely passive. It depends on there being light around. And so that light comes into the eye, and it is refracted, it is bent. So there are a number of materials that light passes through before it gets to the photoreceptor cells on the retina. So you have, for example, the cornea, the very thin layer on the outside of the eye. More importantly for our purposes, you have the pupil, which of course is the hole in the iris, the colored part of the eye. So you have a pupil that we see as dark. And so that pupil can get bigger or smaller. You've probably seen pupils constrict and dilate. You may have gone to the eye doctor and had your pupils dilated. So that makes that hole in the middle of the iris bigger. That's done so the eye doctor can see inside your eye and look at its physical parts. But your eye also does this naturally. So light goes in through the pupil. If it hits the iris, it just bounces off or gets absorbed. So only light that gets in through the pupil is going to be detected by the retina. But before it gets to the retina, it goes through the lens. And the lens, we're used to thinking of lenses as being made of glass or some hard material. The lens of your eye is made of proteins, and it's a little bit squishy. So it can bend, and it bends and deforms based on the contraction of muscles that are attached to it. So muscles will pull on it to make it sort of thinner and flatter, or they'll relax and let the lens sort of bulge and become more circular. And that refraction of light is affected by the shape of that lens. Just like in a camera, the portion of the visual field that's in focus depends on where the lens is relative to the back of the camera, so you can move it in and out. The lens's shape, not its position, it doesn't really move forward and backward, but it is reshaped by these muscles, and that determines what is being focused on out there. So this light is bent, and it is eventually makes its way to the retina. And we'll, again, focus on the retina here in a minute, but this process already is vulnerable. So there are times and circumstances under which it breaks down. So if you are nearsighted or farsighted, or know someone who is, that is a disorder of light refraction. So myopia, nearsightedness, individuals with nearsightedness tend to have an easier time seeing things that are up close, but their distance vision is blurry. And the reason that is that the lens is refracting light, but that light is being focused in front of the retina. Ideally, you want the focus, the sharpest possible image, to be on the retina, but the lens is effectively focusing it in the wrong place. So that's myopia. Hyperopia is the opposite. It's farsightedness, those who can see at a distance, but have trouble with close-up vision. And you can have hyperopia from birth, but most commonly, it's something that happens as you age, and you adjust it by getting reading glasses. People who are nearsighted can adjust for myopia with glasses as well. That way they can see both near and far. So that's all about light refraction. Okay, so that's how light gets to the retina. But let's look at the retina itself. And in this figure from your book, you can see already that the retina is composed of layers of different kinds of cells. We're not going to go into detail on most of these cells. But what's interesting about many of them is they exhibit what we call graded potentials. So from our unit on action potentials and synaptic transmission, we know that action potentials are all or nothing. They either fire or they don't. And if they fire, they are the same size in terms of amplitude, in terms of how much electrical charge is moving down the axon. And we also know that response, that action potential, will occur if threshold is reached. And it doesn't matter if the stimulus just barely reaches threshold, or if it's a really big stimulus and easily passes threshold. The action potential will be the same either way. These cells in the retina are different because that's no longer the case here. They exhibit these graded potentials. So a very strong stimulus will result in a lot of neurotransmitter release. A moderate stimulus will result in a moderate amount of neurotransmitter release. And a weak stimulus will result in a little neurotransmitter release. So the amount of neurotransmitter being put out depends on the strength of the stimulus, and that's something new. So what we call photoreceptor cells, your rods and cones, those are an example of these cells that exhibit graded potentials. You may have heard of rods and cones, and those are cells that we will talk a bit about. The others we're not going to focus on as much. They process information coming from the rods and cones. And these are cells like bipolar cells and horizontal cells. And again, we're not going to dwell on these, but what they have in common is that they all use these graded potentials. They do not fire your standard action potential. They release neurotransmitter based on the strength of the stimulus they're receiving. And your rods and cones are sometimes referred to as the scotopic and photopic systems, respectively. All that really refers to is the conditions under which they respond. So the scotopic system, the prefix on that word, scotopic, comes from the root word for darkness. And so this refers to your rods. And your rods do need some light. They can't work in total darkness, but they don't need much. So if you're in dark conditions, it's your rods that you're really depending on to see the world around you. And that's in contrast to the photopic system. And the photopic system is your cones. And as we'll see, you have different kinds of cones. The cones are what let you see color, which is why you need a certain amount of light to see in color. Otherwise, everything looks sort of gray because you're depending on your rods. So you need more light for the photopic system to work, and more light than the scotopic system does. So the cones need more light than the rods do. And we'll see why that's important later. But here are two more images from your book. And here we have a rod cell and a cone cell. And the structural differences are not very important. But what we see here is that they connect to other cells in the retina. That's what we see here in the lower right. And so they release neurotransmitter. That's what these dots are. They release neurotransmitter just like other neurons do. The difference is how they do it. So they release neurotransmitter just like neurons in the brain, but unlike the neurons in the brain, they can release a lot or a little or just more than none, anywhere in between, because of these graded potentials. So that's the point here. They exhibit these graded potentials. You have rods and cones that work in different light levels. So how does that work? How do these respond to light? So again, we have to get from sensory transduction to, in this case, graded potentials and neurotransmitter release. So the way this works is through a molecule called rhodopsin. And this is a figure from your book. And the important part here is that light hits these rhodopsin molecules, and any of your rods and cones will have lots of these opsin molecules. There are different kinds. The difference is not important. But you have these molecules embedded in these cells, and when they get hit by a photon, they change shape. And that change in shape sets off a chemical cascade, a chemical reaction, that eventually results in closing down sodium channels. So on the right of this figure from your book, you see these sodium channels, and actually they're normally open. But when light hits one of these rhodopsin molecules, it sets off a chain reaction, and a molecule comes along, binds to these channels, and closes them, so sodium no longer flows in. What does that mean? Well, it actually means that when light hits your rods and cones, it actually hyperpolarizes those photoreceptor cells. They release less neurotransmitter. Other cells that exhibit graded potentials will respond strongly to a big stimulus. The rods and cones actually shut down when they get a big stimulus. So it's the opposite of what we usually think of. We think of a stimulus coming in and then a cell responding, and the bigger the stimulus, the more likely it is to respond. Here, the photoreceptor cell is always releasing neurotransmitter. And in fact, it only stops or lessens the amount that it releases when a stimulus comes along, when light comes along and hits these rhodopsin molecules. And that may seem a little backwards. Why is it that the stimulus, light, causes a stoppage in the signal rather than starting the signal? But it doesn't really matter. Either way, the nervous system can tell when a stimulus has arrived because it can tell when these cells become active just as easily as when they become inactive. So it doesn't matter which way the system changes when a stimulus comes in, as long as it does change. So if you record the electrical potential in these photoreceptor cells and you flash some light on them, a little bit of light, a dim light, will hyperpolarize them a little bit and decrease the amount of neurotransmitter they're releasing by a little bit. A bright light will really hyperpolarize those cells and will shut down neurotransmitter release. So again, a little bit the opposite of what we're used to, stimulus coming in and causing neurotransmitter release. Here, these photoreceptor cells, the stimulus, light, comes in and stops neurotransmitter release. But either way, the neighboring cells can tell that there's been something different, that there's been a change in the amount of stimulus being received because the neurotransmitter levels have changed. And that's the important part. And it's not the same across the entire retina in terms of sensitivity to light. So if you look in the middle of the retina, that is the spot in the retina where light falls, that corresponds to the middle of your vision, what you'll notice is that there are more cones there. So those cones are in this spot that we call the fovea, the central part of your visual field that corresponds to the middle of the retina. And there are more cones there. And again, cones are responsible for color vision. They all are also more responsive in bright light. And so if you're in a brightly lit room, and especially a colorful room, you're really using your cones quite heavily. And the middle of your vision is where you have the densest concentration of cones. So if you look here at this diagram from your book, you'll see these sort of different blobs that represent rods and cones. The blue blobs are rods, the grayer blobs are cones. And so in the middle of your visual field, in the fovea, it's almost entirely cones. The further away you move from that middle spot, you'll find more and more rods and fewer cones. So again, the rods and cones respond to different intensities of light. The rods are better in dim conditions, but once it gets bright enough, the rods kind of max out. They can't tell the difference between bright light and really bright light because they're already giving the strongest response they can. Cones, however, can tell the difference between bright light and really bright light. What they can't do is detect light when it's pretty dim. So there you need your rods. But you have an amazing range of light intensity that you can still see in. We're talking about a factor of 1 billion or 10 billion. So the dimmest condition in which you can still see, if you compare that to the brightest condition in which you can still see, the amount of light between the two is different by a factor of 1 billion or 10 billion. So it's huge. So how do we deal with that? How are we able to see in both dim light and bright light with just these two kinds of photoreceptors? Well, one thing we can do is dilate and constrict the pupil. So that can control the amount of light coming in to a certain degree. It can change it by a factor of about 20 from the most constricted to the most dilated diameter of the pupil. And so the more constricted the pupil is, the less light that's getting in, that hole is smaller. So there's less of a chance for light to get through. So dilating the pupil makes it bigger. You get more light coming in. And we know the pupil dilates during our flight or fight response. So we have pupil dilation and constriction. That's one way to control the amount of light coming in. But we also have that concept of range fractionation. So different components of our visual system, again, the rods and cones, they respond differently in dim and bright conditions. So the cones do very well in bright light. They're not very useful in dim conditions. The rods do very well in dim conditions, don't do very well in bright light. So that range is fractionated. Different photoreceptor cells handle different levels of brightness. And then there's adaptation. And you've experienced this if you've ever sat in a movie theater, for example, and sat through an entire movie, and then you walk outside right afterwards, and it's really bright outside. But after a while, it seems normal again. So the cells in your retina adapt, and these photoreceptor cells adapt. So they'll change their sensitivity if they've been in dim or bright conditions for a long time. And so that's why it takes a little while to get used to darkness or to get used to bright conditions. because your photoreceptor cells are actually making more or less of those rhodopsin molecules. And so if they have a whole bunch out there, they're going to be really sensitive. If they don't have very much, they're not going to be as sensitive. But they can shift their sensitivity based on how much light is out there. But it takes a little while to do that, which is why, again, walking from dim to bright or vice versa, you can't really see very well for a little while when you first change the amount of light level that you're experiencing. So these are all ways that our visual system responds to these different intensities of light and handle this billion-fold increase or decrease in light levels. Going back to the structure of the retina, we kind of blow it up a little bit more. We see that the retina, in a sense, is built backwards. What do we mean by that? Well, this diagram from your book, the front of the retina is at the top of this diagram, and the back of the retina, where the photoreceptors are, that's right up against the boundary of the eyeball. So light actually has to go through all of these cells just to get to the photoreceptor cells. And what are these cells? Well, some of these cells are called retinal ganglion cells. We haven't mentioned those yet. But these are cells that do visual processing. But most importantly, their axons form the optic nerve. And the optic nerve is that bundle of axons that goes from your eyeball into the brain. So you've probably heard of the optic nerve. But that's what retinal ganglion cells do, is their axons sort of bundle together and exit the eye. And so they are the last kind of cell that gets used by the retina before that information leaves the eye. What's interesting, though, is that optic nerve, again, is a bundle of axons, but it has to leave somewhere. And there are so many axons that where it exits the eye, there's no space for photoreceptor cells. So you actually have what is called a blind spot, or the optic disc, on part of your retina. It is thankfully not in the middle, where you have your most sensitive vision, your cones are most densely concentrated. It's off toward your nose a little bit. But there is a spot in your eye where the optic nerve leaves, and you don't have any photoreceptors. And it's called the blind spot. So you actually can't process any stimulus that falls there. And yet we don't experience a dark spot or a hole in our visual field. When we look around, we very much see a complete visual picture, despite the fact that we can't actually process information in one particular spot on our retina. So it turns out our brain fills in that spot. It takes its best guess as to what's there based on what's around that spot, and then fills in the missing information. Most of the time, it's pretty accurate, but not always. And there are visual illusions that take advantage of that blind spot. So particular images where your brain fills in the missing information, but fills it in wrong. So here on this diagram, we see the sort of notch in terms of receptors per unit area. So we can see these two curves for rods and cones. This again reinforces the idea that cones are really common in the middle, in the fovea, and rods are more common out toward the periphery, which is why, by the way, if you're ever in dimly lit conditions or you're trying to see a dim object, don't look right at it. Look just off to the side. If you look right at it, that image will fall on your cones, and your cones are not sensitive in dim conditions. If you look just off to the side, then it will fall on your rods, and your rods are more sensitive. You'll be more likely to see the object. But we also see this notch here, this blind spot. And so that's where you have no photoreceptors, because that's where the optic nerve leaves. And the optic nerve, again, is the collected axons of the retinal ganglion cells. So we're not going to talk much more about those cells. They do have a processing role. But for our purposes, their most important function is to form the optic nerve. So we've seen that these photoreceptor cells, these rods and cones, respond to different brightnesses of light, but they also respond to different colors. And color corresponds to the wavelength of light waves that we're receiving. And whether it's the object itself or if there's paint on the object, objects will reflect some wavelengths of light and absorb others. And so it's whatever light waves we're getting, that is, the ones that are reaching the retina, that determine which color we perceive the object as. So that's a property of the object and the illumination of the object. But different photoreceptors will respond to different wavelengths of light. Some we call S cones, so there you have 3 kinds of cones. The S cones, S is for short wavelength, respond really well to bluish light and don't respond as well to yellow light, for example. Whereas M cones respond to medium wavelengths. They respond very strongly to green light, but not so much to blue or red light. And so depending on the combination of response sizes that you're getting from these different cones, you can tell what color of light is hitting them. So that covers both the wavelength that a particular cone is most sensitive to, but also anywhere in between. Because again, you will get different response sizes from the different cones, and you can tell what color it must be based on the combination of those responses. Your rods also respond optimally to a particular wavelength of light, but your rods are all the same. So your rods can't differentiate color, but your cones can, because you have different kinds of cones that are optimized for different wavelengths of light. So your retina can determine what color of light is hitting a particular spot based on the activity level in those photoreceptor cells. But of course, your retina doesn't do all of your visual processing. It does only a very small part of it. So we have to get the information from the retina to visual cortex, and we'll start with primary visual cortex. So visual cortex, of course, is located in your occipital lobe, the lobe of your brain farthest back in your head. But to get there, it has to leave your eyes and go through what's called the optic chiasm. And the chiasm, if you know your Greek letters, or especially if you're affiliated with a fraternity or sorority that has chi in its name, you know the letter chi looks like an X. And that's all that's happening here, is the optic nerves merge and then split again, and it looks like an X. So it's called the optic chiasm. So why do they do that? Well, it turns out that one of your eyes gets information about both the left and right half of your visual world. The other eye does too, obviously. But your brain reorganizes that information so that it isn't split by eye anymore, it's split by visual field. So the optic chiasm remixes that visual information. From the eye, obviously, the optic nerve leaving an eye only has information about that eye. But that information will consist of both left and right visual worlds. We call those visual fields. So the optic chiasm remixes all those signals and then splits again. And that way, information about your left visual field goes to your right visual cortex, and information about your right visual field goes to your left visual cortex. So we've seen this again and again, the contralateral organization. We see a similar thing happening here, but it's not that your left eye is processed by the right side of your brain. It's that your left visual field is processed by the right side of your brain, and vice versa. So that's how information is organized. Before it gets to visual cortex, though, it goes through the thalamus. We know that all of our sensory information, with the exception of our sense of smell, goes through the thalamus. And for vision, it goes through a part of the thalamus called the lateral geniculate nucleus, or LGN. And some processing happens here, not a whole lot, but some. And that information gets processed a little bit and then sent on to primary visual cortex, also called V1, 1 for primary, V for visual. So we have primary visual cortex. And it turns out, this bottom figure is from your book, and it turns out that your primary visual cortex is arranged spatially. So if you show the left part of this diagram, you show that picture to a monkey, for example, and record from its visual cortex, the corresponding part of visual space will be active in that monkey's cortex. So here we have a color-coded version of this image laid out in visual cortex. And it's not circular the way the original image is, but there's still a one-to-one mapping between a location in visual space and a location in primary visual cortex. And that's true for humans as well. Obviously, we don't do this sort of experiment in humans, but visual cortex in humans is laid out spatially. There's an interesting phenomenon here, talking about the lateral geniculate nucleus and the primary visual cortex. So if your primary visual cortex gets damaged, you are no longer consciously aware of your visual world. You can't see it. At least that's the way it seems. However, information is still hitting your eyes. It's still hitting your retina. It's getting to the lateral geniculate nucleus, and it turns out there's sort of a shortcut, sort of an alternate route from your eyes into your brain that doesn't use primary visual cortex. And these patients have what we call blind sight. So they say they can't see, they are not consciously aware of their visual world. But for example, if they are forced to guess what an object is, they are right a surprising amount of the time. If you have a slit in a piece of paper and you give them a coin and say, orient that coin so that it fits into the slot, they'll say they can't do it. They say they can't, that they don't know which way that slot is, if it's horizontal or vertical. But if you tell them just go ahead and try, then they're able to orient that coin into the right orientation to fit through the slot. So they're not consciously aware of their visual world, but clearly some information is getting in because they can still respond to these questions accurately if asked to do so. So a very interesting phenomenon, and it shows there are multiple pathways into the brain when it comes to visual information. Okay, so we can see light, we can process color, but of course that's not all we do, and we don't experience the world in terms of blobs of color or blobs of bright and darker shades of light. We have, for example, what's called binocular vision. There's a similar figure in your book. I like this one better, so I've used it here. Different animals have different degrees of what we call binocular vision. Binocular vision is one of the critical components in depth perception. You can see how far away something is by using both eyes and comparing the images that are coming into both eyes. So as a demo, if, for example, you hold up your finger and you hold it close to your face and you look at it with either your left or your right eye, you see different images. If you, for example, with one eye, you'll be able to see the fingernail or most of it. On the other eye, you can't really see much of it. If you hold your finger farther away, you're getting pretty much the same image for both eyes. And so your brain uses that difference to tell how far away an object is. So here, this diagram, again, not from your book, but you have a similar one. On the left, we have a rabbit. And the rabbit's eyes are on the sides of its head. There's very little overlap in front of the rabbit in terms of things that both eyes can see. On the right, we have a cat. The cat's eyes are on the front of its head. And so there's a lot of its visual field that's picked up by both eyes. And the same is true of us. A lot of our visual field we can see with both eyes. It's only on the extreme sides that we only see with one eye or the other because our nose gets in the way. And the difference here between a rabbit and a cat is that one is a prey and one is a predator. So if you are a prey, that is, if animals tend to eat you rather than you eating others, it's most important to you to see that predator first and run away. How far that predator isn't as important. As soon as you see one, run. For a predator like a cat, it has to pounce, and it has to pounce the right distance. So it needs to know how far away that prey is. So for a predator, the distance to the target is very important. For a prey animal, just the fact of it being there is the most important part. So evolutionarily, rabbits have eyes on the sides of their head so they can see the most of their visual world. They sacrifice overlap, binocular vision, for the sake of seeing more things out there. And again, for predators, and we obviously fall in the category, our depth of perception is very important. We need to be able to see our target and see how far away it is. And we also depend heavily on our visual cortex. We depend heavily on our visual sense. So about a third of our cortex is taken up by visual processing because vision is so important to us. And we've talked about primary visual cortex, but we have different areas in our visual cortex that do different things. And these are roughly split up into two pathways or streams. You have the ventral stream, and we know from our unit on neuroanatomy that ventral means the bottom part of the brain. So this goes from your occipital lobe down toward your temporal lobe. That's what we call the what stream. So the areas that process visual stimuli in this ventral stream identify the object, tell what shape it is, what color is it, what is the object itself, what do we call that? The more dorsal areas, the ones that run along the back and top of the brain, that's the where stream. So it determines where an object is in your visual field, and also whether that object is moving. Where is it moving to? And so that's more about not what the object is, but where it is. So we have this distinction between the ventral pathway or ventral stream and the dorsal pathway or the dorsal stream. And it's the difference between what and where. And again, we have rods and cones along the back of our retina, and those rods and cones correspond to a particular location in visual space. But it doesn't just stop there. We don't experience the world as collections of dots, which is really what our retina does, but our photoreceptors have. All they can tell, any given rod or cone, all it can do is say whether it's being stimulated or not, and how strongly it's being stimulated. It can't say anything about nearby photoreceptors, but other cells in your retina and your brain can. So we have to go back to the concept of receptive fields. We've already seen this for the somatosensory system, but a particular neuron in your visual system will collect input from various photoreceptors, and it will respond most strongly when just the central photoreceptors are active, or alternatively, when just the surrounding ones. So you go back to these donut-shaped receptive fields. And so we're starting to get more complex processing capability. It's not just whether one photoreceptor is active or not, it's the shape. What's the shape of the stimulus that's hitting this receptive field? Is it just the middle part? Is it just the surrounding part? Is it both? Is it neither? And so now we're starting to tell something about shape. And so, for example, what this just lets us do is build up these receptive fields and combine them into things like lines. And of course, a line has an orientation, has an angle. It could be going straight up and down vertically. It could be going side to side horizontally. It could be tilted at an angle. And so you have cells in your primary visual cortex, V1, that exhibit what we call orientation selectivity. So if there's a line out there, if it's tilted at a certain angle, a given cell will respond really strongly. It sort of likes that angle of line. If you rotate that line, it doesn't respond anymore. And so you have orientation selectivity, that is, cells that fire more or less depending on the angle of that line. How does this work? Well, it works by combining the receptive fields of various neurons, various photoreceptors, for example. And so this is an example not from your book, but I happen to like this diagram. If you combine a bunch of these receptive fields and they're all next to one another, they will form a particular shape. And so in order to hit the central part of each receptive field, what shape does that? Well, it's a line that's exhibiting an angle that crosses all of those middle receptive fields. And so only that shape will get the biggest response because, again, those earlier cells have a donut-shaped receptive field. They respond most strongly when a stimulus is in the middle and not on the surrounding part. So a stimulus that hits just the middles is going to activate that later neuron most strongly. So it selects lines of that orientation. It responds most strongly to them. So now we're getting even more complex. Not just these donut-shaped receptive fields, but actual shapes and shapes at certain angles. You can also exhibit what's called motion selectivity. And this is an area called V5. Suffice it to say, it's not V1, it's a different area, but still part of visual cortex. So let's say you have a bunch of neurons, and each one has its own receptive field. It responds when there's a stimulus in its field. And you can have different neurons that have overlapping receptive fields. So we'll call them A, B, C, D, and E. So neuron A responds really strongly when there's a stimulus far to the left. B is most responsive to stimuli that are left, but not as far as A's stimuli. C is right in the middle. D responds to a stimulus that's a little bit to the right of middle. And E responds to a stimulus that's way out there on the right. So you can combine the outputs of these neurons. You can have a neuron that listens to all of them, and it can be selective for objects moving at a particular speed. How does it do this? Well, if we imagine that A, B, C, D, and E, that each one has a certain response time. So A, for example, takes the longest to respond, or takes the longest for its signal to get to our neuron that's collecting those responses. B doesn't take quite as long as A. C doesn't take quite as long as B. D doesn't take as long as C, and E is the quickest. It responds almost immediately. So if this middle neuron, if this neuron that's listening to all of its neighbors, if it adds up all these signals, well, a stimulus that hits A first, and then B, and then C, and then D, and then E is going to produce its maximal response. Why? Because again, A gets stimulated first, but its response takes the longest. B gets stimulated second, and its response takes a little bit less time than A's does, and then so on. So all of these responses will end up hitting our listening neuron at the same time. And that response, when added together, is going to hit threshold. So you'll have a neuron that's listening to these neighbors, gets their signals in a particular order, adds them up, and then says, hey, there's a stimulus moving from left to right. It's hitting A, and then B, and then C, and then D, and then E. And you can contrast that with the stimulus moving in the opposite direction. It hits E first. Well, E is the quickest to respond, so that stimulus goes immediately. Then it hits D, and then C, and then B, and then A. A's response takes the longest. So what do? You end up spreading out that stimulus across time. So this listening neuron gets all of these signals, but they're all spread out across time because it's not the order in which that neuron prefers those stimuli. So you spread out those stimuli, those responses from those neighbors, you add them all together and you don't get anything. It's that first version that hits A and then B, C, D, E. All those signals end up arriving at the same time, and you get that big response. Whereas in the opposite direction, you don't. So now you have neurons that can respond to not just motion, but motion in a particular direction. And that's what area V5 does. Now, this is a diagram not from your book. We see, again, this dorsal pathway and ventral pathway, otherwise known as the dorsal stream and the ventral stream. And you have multiple areas in visual cortex. The point here is not to memorize these areas, just know and appreciate how many there are. And these areas process things like shape, orientation, color, movement, identity of the object. And other areas do different things as well. So you have area V2, for example, and V2 tries to figure out what shape things are. So you get what's called a perceptual fill-in. So if you look at this diagram from your book, what you probably see is a white triangle in the middle that's overlapping another triangle and three circles. Literally what's here are sort of three thin V-shaped lines and three sort of Pac-Man shaped objects. But that's not what your visual cortex probably interprets the stimulus as. So again, V2 tries to figure out the simplest explanation for what shapes are out there. And in this case, you probably see a white triangle sitting on top of simple shapes rather than a collection of 6 complex shapes. Similarly, if you look at this picture, this is not from your book, but if you either download these slides or just look at this image in this video, if you look at these shapes, you probably don't see much. It looks like a jumble of random shapes. If you look at this, I'll give you a minute to look at it. Try and figure out what those light-colored shapes are. And you can probably do it. Because this image is added, it's the exact same light-colored shapes that are on the left. But this image has added this sort of abstract blob shape on top of it. And so if you look at the shapes on the right, you can probably see that the light-colored shapes are the letter B. They're capital Bs, oriented different ways and sort of jumbled. But what's going on here is that dark shape indicates to your visual cortex that something is covering up these shapes, these lighter shapes. And so your brain tries to figure out what those lighter shapes are, and it doesn't. It says these capital Bs. On the left, you don't have this blobby abstract shape to give you a clue as to what's happening. It just looks like a bunch of random shapes. But on the right, you know that these are bigger shapes and that part of them has been covered up. And so your visual cortex is able to determine that these are capital Bs being covered up by a closer shape. So that's another form of perceptual filling. And again, we don't experience the world as a collection of dots or even lines. We experience objects. But this process can go wrong. You can experience what's called agnosia, which is a disorder in which you have trouble recognizing objects. There are forms of this. There's, for example, apperceptive agnosia, where you cannot tell the basic components of a shape. So this is a figure from a different book, but if you give people even simple shapes like a circle or a square or a diamond or the number 3 or 4, if you ask them what these shapes look like, they cannot describe their basic parts. If you ask them to draw these shapes, they cannot do it, even though these are simple shapes. If you ask them to name the shapes, they still can. So they have lost the ability to break shapes down into their small parts and to reproduce these shapes. They still have the ability to name them. And that's different from associative agnosia. Associative agnosia, you can give people pretty complex shapes. Here from this figure, give them a tea bag or a ring or a pen, and you say, okay, can you draw this? And they can draw it. They can break these things down into their component shapes, reproduce the shapes, but they can't name them. They say, I don't know what this is. Or they'll say, oh, that seems familiar, I just can't name it. So this occurs when there's damage to two different parts of the ventral stream or the ventral pathway, that what pathway. So if it's an area closer to an occipital lobe, you have trouble recognizing the basic components. If it's an area farther down into the temporal lobe, you can recognize the basic components, but you can't assign a meaning to those shapes. You can't name them. And so that, again, is the ventral stream. It goes from primary visual cortex down along the underside of the occipital lobe and into the temporal lobe. So depending on what stage in that process you disrupt, you either can't recognize the basic elements, but you can name it, give it a complex meaning, or you can't give it a complex meaning, but you can recognize the basic elements. And agnosia can come in very specific forms. You can have what's called prosopagnosia, which is an inability to recognize faces. So it turns out there are special areas in your brain for recognizing faces. And people with prosopagnosia often don't know it because we're so seldom presented with just a picture of a face. We often have the person's voice or the way the person moves or a video of the face moving, and you can tell from a facial expression, perhaps, or from the way they're dressed. But if you're given just a still image of the face, if you have prosopagnosia, you cannot identify that face. You don't know who it is. You have to wait until they speak, until you see more of their hairstyle or their body, for example. And if you look at a monkey at a particular part of the temporal lobe, there is an area that responds very strongly. So the top part of this image, you see exponentials, those spikes that are being added together. And the bottom part is the visual stimulus the monkey is getting. So it's a monkey face. And these neurons respond very strongly to monkey faces. They respond less strongly if you remove parts of the face. So that's not a very natural stimulus, the monkey face with the mouth removed. And so they don't respond as strongly. They also don't respond as strongly if it's a human face. whereas the equivalent area in the human brain would respond very strongly to human faces, not as strongly to monkey faces. In a monkey, if you keep the same components, all the parts are there, you haven't wiped anything out, but you've rearranged it. So now there's an eye at the bottom and part of the mouth is up top. If you rearrange it that way, you get some response, but not a very strong one because it's not a face in its usual geometric arrangement. And so that's a very complex stimulus, but faces are very important stimuli. We use it to identify people in our group. We use it to identify emotional expressions. And if you, for example, show a hand, which is another body part and an important one, you still don't get a response, or at least not a very strong one. Hands are important, but this area is specialized for faces. And visual cortex can change, and we see that as a theme over and over again in this course, that neurons are not set in stone, so to speak. So you can learn, and as you learn, your visual system will devote more cells to particular kinds of visual stimuli. So this is a diagram from your book. These are some complex visual stimuli, sort of circular spiral shapes. And different neurons will respond most strongly to different kinds of this shape, or different thicknesses of spiral, or different colors, or different orientations, rotationally speaking. And these are some other shapes. And these are more complex. They're combinations of shapes. But if you give a monkey rewards for accurately identifying these shapes, it will devote more of its visual cortex to specializing in these shapes. And the monkey becomes very accurate at identifying these shapes in particular and telling the difference between these shapes and shapes that are very similar but not quite the same. So the visual cortex can change, it can learn. And of course, damage can occur in the visual cortex. So if you get part of your visual cortex that's damaged, or part of your retina that's damaged, then you can exhibit some recovery. So using the example of the retina, if you have a lesion, a damaged spot in part of the retina, you're no longer going to get visual information for that part of your visual world. So now those corresponding cells in your visual cortex don't have anything to do. However, the nearby areas in your brain will sort of move in and take over. And so you'll be able to process nearby areas in visual field, perhaps more accurately. So you don't get that part of your visual world back, but you do get processing of nearby areas in the part of the visual cortex that now has nothing to do anymore. So your brain can adapt. It can adapt based on learning. It can adapt based on damage to the brain or the retina. So once again, evidence that our brains are not written in stone. They can change over time based on need. Okay, that will do it for our unit on vision. So be sure to take the online quiz as always. And for next time, read chapters 2 and 3, not all of them. So look at the syllabus for the reading. We'll be talking about research methods. So how do we collect data on the brain? How does that process work? What can we do with those methods? What can we not do with those methods? What are the risks and pros and cons? And as always, do the reading before lecture. The lecture will make more sense to you. You'll be able to put the content into context. So we will cover research methods next time, and I will see you then.